Okay, so I'm going to try to make like what the the steps of the procedure look like, just because I don't know if visualizing it might help you guys, and also if I can explain it, then I know um that it helps me figure it out. Okay, so the first thing we have to do is preparing everything before leaving school. And the first thing is preparing the the hydrochloric acid solution, which we already have, which is just 2 moles, 2 mole, which is pretty simple. So my imaginary hydrochloric solution is red. Um, I also have my 5 gallon bucket. I know, huge. This is my sink because I don't have an actual sink here. I do. It's not here. Anyways, okay. So based on what we have from Rivers, is pretty much to first open the bucket, which is my imaginary water. This is just tap water because I'm not wasting real water. Take it, rinse it. Obviously, in real life, we don't have to put the cap back on. Rinse it, throw it to the sink. After that, we make our solution, and obviously it's gonna be mixed with the already mixed with the water. So pretend I added the solution from the water. If that makes sense, like that our solution. This is our already made solution. I'm scared of these. See, because I read it to be 500 milliliters. I don't know if that's gonna be enough, but honestly, you can always just change it. Okay, and we make sure that it touches every part of it. Once it's clean, we dispose of it in the this. And then rinse everything out. Make sure nothing stays. And that should not be this hard. And once it is fully washed out, we put it as to let it dry, but upside down, to make sure that no bacteria or airborne contaminants get in. So now we're gonna leave our little bucket to dry upside down. Also, I think we should make sure that the lid is also cleaned. Clean the lid, and also leave dry upside down. And our thingy. Then what was after was, hold on, where's my thing? Make sure we have we have our pH probe, which should be already calibrated. Here's my imaginary pH probe, Also, all of our testing kits, which would be the pH one, all the oxygen, salt, nitrogen, and phosphate. These will be placed in the bag they give us because I feel like it's the safest place to put them. And we'll place all of them there. Also, we have to take our little, sample water dipper thingy, and our plankton net thingy comes with a little bottle you put at the bottom, so that when the water passes through, it catches the phytoplankton. And once everything is done and our bucket is dry, just close it, and we take it to whichever car we're taking. So That part was pretty easy, but I just want to make sure I do everything for the sake of making everything. So that's the before going to the site. Okay, so once we're at the site, we would take all our materials, all of them would be here, and we would set them out to make sure we have everything. And because all tests have to be done pretty much at the same time to make sure that time is not a factor, because especially because it's flowing water, even those the flowing water can change the measurements, I think, and plus it's science, it's kind of how it goes. But once we have everything laid down, we write it down, like, data, whatever which is color of the water, soil / sand color around, any smells we might see, the time of the day, weather, temperature, when is last rain, and any other noticeable like things that might be important later on. I also think we should try to keep pictures, just a log of pictures, like, hey, so we didn't think about this, but we go back, we noticed that there was X and Y, and they all had it, and because we might not notice, so just keep a log of pictures. So technically, in my scenario, this is my watershed where we're testing. Hold on. This is not the best book for this. So we have our watershed. Hold on. There we go. We have the watershed with, this is, natural water, which, for the sake of differentiability, I'm gonna color a different color. That color being green, because my favorite color, obviously. Okay. So here we have our hypothetical watershed. I know you can't see it, but it makes sense. So the first thing we're gonna do is take the pH probe, measure it however it goes. Like, I didn't look into it. I'm so confused about it. Wait, I don't even know if we should get it. And then, anyways, we will take a thermometer, measure the water. When we do it, it should be kept in the water for at least a minute to make sure it acclimates and make sure that the reading is correct. And we should probably take it out, dry it, and make the measurement at least 3 times, so that make sure it's constant and it's not just a weird variable or it didn't measure correctly. After that, we're gonna take our bucket. We have our bucket, and we're gonna take the water dipper. We're gonna get our water samples. We're gonna add them to our, our 5-gallon bucket. I don't know how much we're gonna need, but hypothetical, that's it. And that way it makes sure that all the tests we make are constant because it's the same water. And with this water is where we would test the pH, the dissolved oxygen, salinity, phosphate, nitrite, etc. The only thing I'm not quite sure of is whether dissolved oxygen should be tested here, or, well, I mean, we are testing it here, but I think we should also test it in the natural environment, because the way that, at least what our research is, that there's less dissolved oxygen at the bottom, and the less there is, especially 3 parts per million or less, it's pretty much no life. So if we go test and we see those low numbers. That's something to keep in mind. While it is kind of normal to have lower levels at the bottom, it should never go anywhere near 1. So that would be something interesting if it did were to happen. And after that, after we make sure all tests have been done, we seal it, and we take our cooler, hypothetical cooler, and put it here, and just take it to school, pretty much. And that would be that part of it. For the next one, I can't actually use it, but it would be for the cytoplankton, the plankton. I can't say that word. We would take the net, the one that I put in the list. It's the net, and it has a 125 milliliter jar thingy that goes in the bottom. You screw it, and you will dip it in the water, back, like push it inside and out for a little bit, and then keep it as at a 50-degree angle to make sure that the water's flowing through. If there's not enough current, you will just like walk with it to make sure it stays flowing for 3 to 5 minutes. After it's done, you're just gonna like dip in the water a little again and again to make sure that any phytoplankton that might have stayed here goes inside. After that, they're still gonna be together, and you're gonna take those like wash, like the bottles that we use for titrations to clean the tips. We're gonna use that around, and then we unscrew. And usually, because a lot of people, I don't know why they did it in that video, but a lot of them that I watched, they would use the Lugol's the Lugol solution almost immediately, but because we're doing it in lab, we would have to move this to an amber like, how did I, Jesus Christ, I forget words. Okay, so once this finish being taken, we clean it with the bottle, we take it, and to make sure that the phytoplanktons are preserved, we're going to change it into an amber glass bottle, which, because it's a lot more opaque and also glass instead of plastic, it preserves the phytoplankton, and those should not be kept more than 24 hours. So we would move it and place it also on the amber glass. Example, for pretend this is the amber glass bottle, just also place it in the cooler, and then make sure all materials are in the bag, in the car. Make sure this is in the car, and just that's the end of insight part of the procedure. Okay, so once we get back to school, we were gonna take our 5-gallon bucket, and I made it to be 8 deli samples, but I only have 3 because it's a tiny sample, obviously. So we take, we take the samples, we divide it into how many deli containers we consider to be the best amount. In the procedure as of right now is written as 8, sorry. Once we have our containers all full, preferably to the same amount, which will completely depend on how big the deli containers are. These obviously will seal, and we will take those and place those into the freezer. Because obviously, if we're able to continue testing that same day, I would consider it better. However, because I'm not sure how we're allowed to leave the school, if we're gonna allowed to leave and it'll be too late to do testing, I would just say put them in the fridge and just start fresh the next day to make sure that we have time to do anything that we have to do and make sure everything is done at the same time. So forward to the procedure for lab testing. We, for the, how it is, place 3 deli containers at room temperature for about 1 hour. So just get to school, place them outside, and so they're mostly liquid. And I made it as to transfer the samples into test tubes depending on how many tests we plan to do. I was thinking about it, and I don't think it might have been necessary. I was thinking originally about it because of the water bath. They can be plastic, but we can always transfer the samples to a glass bottle and just place that the big glass, like the beakers, and just transfer the samples to the beakers, and then use those to warm them up in the water bath, which I think might be the best. And just make sure that they stay for 10 to 15 minutes or until the water is fully defrosted, but just make sure that they do not go above room temperature. And we would go back to the notebook and just, write down anything that might be differences in water color, texture, smell, or any other noticeable things that we might not think about now but might be noticeable. And again, pictures. Just make sure that we have a picture log of everything we do. Then of each deli container will not be mixed. So if it was put in this container, it will be kept in. If it's a different beaker, then there's 3 different beakers. And so for the sake of stuff, let's pretend these are glass beakers that we place in the water baths. We will retest pH, of oxygen, nitrogen, salinity, and phosphate to make sure that there's consistency, and make sure there's no statistically significant differences between what we tested in site or at the site versus in school. That's why we're going to have multiple deli containers, because if, let's say that we tested and it had a pH of 7.4 and we come back, it has a pH of 5, then we can't tell. We won't, that's something happened to the sample. So in that, for example, if this was messed up, then we would just throw it away because it's not usable, because it might have been contaminated or improperly stored. So this goes down the drain. I think, just make sure with Miss McDonald that they are allowed to go down the drain, but I'm assuming so. So that's for all inorganic substances. Also, if there's any inorganic substances we did not test at site, we test them now, and we also write down why we did not test at site, since all of them should be able to be done in the site. And yeah, that's for inorganic substances or abiotic factors. For the E. coli testing, I'm pretty sure we all of us have done like preparing agar plates, just putting down, warming up the agar, pouring it, letting it cool down. We've done all of that. And so we're gonna leave it, and we're gonna prepare the agar and just let it sit for about 24 hours to make sure that there's no contamination. The way I think Miss McDonald does it is that she leaves them outside of the incubator. I don't know if it would be beneficial to put them in the incubator to make sure that nothing grows, to make sure that there was no anything contaminated prior to the testing. So we're gonna put a name for each petri dish. I literally wrote Capstone Group 2 ATG, but whatever you guys, if you guys don't like that, then that's fine. And then we will take the sterile inoculation loop, inoculation loop, and streak it. That's the way I have it set up in the procedure. That is called T-streak, obviously, because it's a T. If there is another version that Miss McDonald would like best, then we will do that, but it has to be kept constant. So I think that the same person should streak, so either me or Janai, because I know Rufus doesn't do. I made it to be 6 agar plates to make sure that, because usually 3 is to make sure that you can find the average, but I want to have extra just in case there's contamination or mishandling of anything, because it's bacteria. If they stay in the water, they're going to multiply more than they would have normally been. So it's going to affect our results. So 6, I think, is the best to make sure that the amount stays the same and we prevent any loss of, well, you know what I mean. We will leave them in the incubator for 24 to 48 hours, depending on time. I would imagine that 24 hours is enough, usually what we do. And we're using something, EMB, emethyl methylene blue agar. It's made so that E. coli, which is lactose fermenter, will show up as either black or a dark center with transparent outer margin, and any other non-lactose fermenter bacteria will just be transparent. But the more lactose it uses, the darker it's going to be, and because E. coli is one of the strongest lactose fermenters, then it will be the darkest, so it'll be easier to observe. And if we do end up having the inverted microscope, we are able to put those plates directly to the microscope and observe the plate, and even be able to identify some of the actual species versus just E. coli or E. coli type X and Y. Obviously, we're going to count colonies, and then I would recommend placing the plates back in the incubator in case anything goes wrong with our data, but I don't think anything should go wrong, so I don't know what we normally do with those. For the phytoplankton, yes, that one, we have to prepare something called a Lugol solution, which is pretty much like potassium and water and a few other things, which is written down on my research paper. It is a very dark color, so it pretty much turns everything brown, but it also weighs down the phytoplankton, so they go down, so it's easier to observe, because the way the inverted microscope works, or actually, let me go through the steps. That's what I'm doing. Okay, so you're gonna take your sample. This is our hypothetical amber glass bottle with our sample. We're gonna take this amber bottle. By the way, before that, the amber bottles we have is 4 ounces, and I don't remember how much that was in milliliters. I think it was 118 something, but I did the math, and that means that for every 100 milliliters you use 1.5, so it ends up being 2 milliliters of the acidic Lugol solution, and you take that solution and you add it to the sample. And obviously for that we would have to use a graduated cylinder to measure it out exactly. Then this would be closed and shaken, just more or less to make sure that it mixes up evenly. And then you're gonna let that mixture rest. Just push to the side. Then after that you have to set up the settling chamber, which is pretty much a line with like a little one of these in the middle to but it has a cap. After you set it up, you add 5 milliliters of this to the settling chamber. So if this was a settling chamber, you'd measure out 5 milliliters with a graduated cylinder, pass the 5 milliliters. That was exactly 5 milliliters. And then this one is covered and is left for 24 to 48 hours to settle, because it's pushing all this phytoplankton from all around the water to the very bottom. Once the time is up, just remove it, and this cannot be shaken, so just like be careful when we take it. And After that, you're going to take a pipette and pipette almost all the water out, leaving no more than 3 to 5 millimeters of, 2 to 3 millimeters of water above. Because the way that an inverted microscope works is that you have a normal one, you have the light at the bottom and the looking at, you see from the top. That's where you look. And it is inverted, so the light's from the top. And so you're able to see what's floating in the bottom. And because the Lugo solution makes the phytoplankton heavy, it pushes all of them to the bottom, and you're able to, shining the light, see the specimen and observe it. From there, we would count all the um, all the plankton. I linked about 2 or 3 phytoplankton identification guides so we're able to identify species, and based on the amount we see of each, just calculate the average amount that we may find. And I don't think we are able to isolate those, but we can probably find information about how they affect the water based on which we find. But if not, that's fine too. Just a thought. And I think those are all the procedures. Yeah, that's pretty much all of them.